I will welcome to the stage Dr. Tim Murphy, who's joining us from UBC, across the other side of town. Tim is a professor in psychiatry and is the lead of the dynamic brain circuits group in health and disease at the Center for Brain Health at UBC. So Tim has done with his lab an extensive amount of work, really impressive work in the area of stroke recovery, particularly looking at high resolution imaging of neural circuits to infer structure and function relationships. And we've done a lot of work too to build those models of different neurological diseases. And so he will talk to us about one component of that work today, looking at automated techniques to model brain and behavior connections in the mouse cortex. And it's like he will focus his talk on stroke.
Well, thank you so much for the opportunity to come downtown to SFU. As I age, it's a little bit better than going up the hill to the east campus, which I've done in the past. Some things. So I'm going to talk about automated techniques to model brain and behavior connections in mouse cortex, and our lab focuses on stroke damage and stroke recovery. I'm not going to talk that much about stroke. We're developing methods to apply them to stroke. I'll mostly talk about automation of brain imaging in mice, and then some approaches using synthetic data to sort of leverage the training of machine learning models. So why do we want to do this automated imaging, and why do we want to image a lot of the brain at the same time? And I think this is really an outstanding opportunity to just see Olaf's talk and the work of Randy and others, as we heard over the last 30 years, really thinking of the brain as we should, as a large interconnected network. And so none of these areas are working completely independently. And this is a slide from a review article by Noni on diaschesis. So we think of the normal brain, we've got the interconnected areas working together. And then when something bad happens, like a stroke, we of course lose activity where we have a lesion, but then we have a lot of interesting things that can happen in connected areas. So I think studying these wide-scale connections in a dynamic manner, we want to do it in mice as the mice are behaving, is something we really need to do to understand the system, because we could we've observed, if we make a stroke on the left side, we also observe significant depression in activity in the contralateral side. There are even times when you can see increases in activity on the contralateral side very acutely due to sort of disinhibition phenomenon. So large field of view imaging is what we need. And so we've been developing paradigms in mice where we can capture the behavior and then also capture the brain imaging at the same time, and do this from multiple perspectives. And this is a project of Dongsheng Shao. And this is a mouse that's trained to reach for water drop. And so what the lab is doing is we're developing methods to really interact with the behavior and the brain activity on a sort of a millisecond time scale as the mouse is doing these types of tasks. And this was a project, this was actually done by Brandon Forrest, who's in the audience here, who was undergrad working with me. And Brandon and Dongsheng developed a real-time tracking system for mice. So this is a post-hoc tracking, but they have a real-time neural network-based tracking of the behavior of the paw, and then we can and we've developed a system to give feedback to the mice as they're reaching. So to reward them for good reaches. And we also have a system to measure brain activity. So two places where we're using closed loop systems in mice to closed loop for the behavior, for the water reaching, and then also closed loop independent experiments based on the brain activity. And this is all work in progress. So I'm just sort of presenting it as preliminary data. Now, things like closed loop analysis, I mean, you need a lot of data, you need a lot of subjects, a lot of time. So we've also been investing in automation of mouse brain imaging, and this is autonomous brain imaging. These are mice that image their own brains 24/7. So they're trained to in a home cage. They get their water, so they're water restricted, and they get their water through a task. They go into a tunnel and they get transiently immobilized. We image their brain, they task, they get water, they're released, and this goes on all day. And the mice are healthy. So these are the mice. They need to be immobilized. This isn't freely moving imaging. So we have there's a small titanium bar here, and then a transcranial window. So we call this mesoscopic wide field imaging. It has about 70 micron resolution. Now that's not good enough for looking at an individual neuron, but we can look at regional activation. As you saw in the previous video, you can see those kind of waves of activity. So here's a mouse that's going to, we call it head fixation, when they come in and they kind of dock, and this blue light comes on. That's to excite the GCAMP. So GCAMP is a genetically coded calcium sensor, way of doing functional imaging from specific neurons. So you can get a lot of data with this type of method. These rows are just sort of thumbnail images. Each one is two hemispheres. The labeling is just in the cortex. And so you'll see these are a series of go trials, no-go trials. You can get hundreds of trials and then intersperse these go and no-go, and then have the mice do potentially complex tasks. And a number of other groups are going in this direction too, to have large numbers of subjects, automated imaging, really take the investigator out of the question, the kind of the equation, reduce variability. So I mentioned this mesoscale wide field, sort of 70 micron resolution. A neuron is 10 microns across. So if you want to look at single neuron coding, we've very recently built, we haven't published anything on it, based on a design from Spencer Smith in Santa Barbara. And this is a 2-photon mesoscope. So this is a 2-photon microscope that has a 7mm field of view. Now, for people that work in humans, 7 millimeters doesn't sound too impressive, but it's almost the width of the mouse cortex. So this is one of our windows. So we've made a window. You have to make a window over the bone, and then we can pick out, these are neurons that we've segmented, and then these are sort of time plots showing clusters of activity. And so this red area here gives you an idea of what we can image. And so we can also move that and image both hemispheres at once. So there's potential to have very wide field of view cellular imaging. This can also be automated. Most 2-photon objectives are water immersion. This is really the only design that we know of with high resolution that's air immersion. So we don't have the added complexity. I don't know if anyone's ever messed around with one of these things, but you have to add a little water drop, or you basically don't get any signal. So it's not required. We also look at social interaction. We're imaging multiple animals at once. I'll just looking at cross correlations between animals. This is a short video clip. We're looking at 2 brains below the GCAMP functional imaging. We bring a second mouse in. we call it a constrained social interaction, and you'll see a lot of activity here in a second. We're trying to understand how animals interact socially. We have a system now where 3 mice interact with a rail-based system, and then so we can very rapidly switch between different partner brain interactions. We're also doing electrophysiology at the same time as we're doing imaging. We're using the high-density neuropixel probes. And in this experiment, Edward Yan is looking at connectivity relationships between the cerebellum and the cortex. So I guess it's kind of like windowing. It's a sort of a spike-triggered method, looks at spiking in the cerebellum, and then how particular cerebellar units, how they're related, sort of the correlation between these units and these wide-scale bihemispheric cortical maps. So we find that there's a very defined mapping between cerebellum and the cortex. And cerebellar neurons that tend to fire together tend to have cross correlations with the same cortical regions. So what we're hoping to do is to have systems where we can record from the cortex in sort of automated fashion, but also using electrophysiology, record from subcortical areas. And sort of our thesis is that we feel that there are going to be strong kind of obligate connectivity relationships between all these areas, and that maybe we could come up with closed loop schemes of conditioning activity in the cortex to potentially affect subcortical sites. We don't have data on this yet, but that's a goal. And these are maps based on, so this is so we've made a spike triggered map. So this is recording spikes in the hippocampus, and then we average cortical activity, and we find a nice connection with the retrosplenial cortex. So we can see these connectivity relationships. So just a few quick slides. So during COVID, we were shut down, and we couldn't do live mouse experiments. So we started to do some synthetic animal work, and we created Luis Bolanos, who is also undergrad in computer science. He's now a graduate student in computer science. He's working with Helga Roden at UBC, computer vision specialist, outstanding young faculty member. But in my lab, Luis had made a model of a mouse in Blender, which is an open source animation program. And we used it to create training data for something called Deep Lab Cut, which is a pose estimation tool. And we were able to publish this. We were really excited. This is the mouse model. It's made from actual mouse CT scans. It doesn't have real bones. It has these rigging kind of actuators in it, and we can control each joint with software. So we added fur, but the thing here, it could also be posed to do things, but it really looked like a fake mouse. So what we did is we then used GAN. We used the kind of deep fakes method, make our fake mouse look real. And then to present to you guys, you probably all know what GANs are. These are generative adversarial networks. And then we can transfer the style of a real mouse to our fake mouse. And so this allowed us, so this is the top here is our fake mouse in an animated pose. And then we used a GAN, what's called a You Got It model. And we transferred the style of some videos that we had of mice in the lab. And so we have the fake mouse pose, so the fake pose, the very defined motor movements, and then we add the shading and everything of the real mouse. And so you can also add I guess things that you would have in the lab that would be imperfect, inhomogeneity and lighting, some small artifacts. So you can use these generative models to create sort of a very defined training data that has the same characteristics as real data. We then used it to train these pose estimators. This is something called deep lab cut, which would allow us to then define positions of paw and particular joints. One thing that's cool with this synthetic animal is we can place these labels. We build the labels into the animation, so no one ever has to click. We know exactly where the digits are in the model. We can also label internal structures of the animal, which would be impossible for a person to do. So we're exploiting synthetic data. We've gone a little bit into direction of human models, and we found there's a huge literature on this, which is great, and people like Michael Black are leaders. And one tool we want to generate based on the human literature, within the human literature, you can go from a 2D image to a kind of a 3D model of a human, or sort of a 3D rendering fairly easily. So we want to build sort of automatic 3D animation of mice, and that's one of our goals now. And so this is some of this is our target, and then we can manipulate our model until we match that particular image. So we're building this kind of differentiatable 3D mouse that we can collect behavioral data into. We also think synthetic models would be a great tool for really any human behavior. We like the idea of capturing human pose using a synthetic model. And this is a paper that we have that just came out in the Journal of Parkinson's Disease. I guess it's still in press. And so we found it very difficult to get any open source video data of patients. I guess that's probably a good thing, but there was this one lonely subject in the Parkinson's literature, and we were able to then digitize their form into this synthetic model. We think this may be a way of making this behavior, this movement data, more shareable, because now it becomes anonymized and de-identified in this synthetic format. So I probably went over time. So these are some of our lab projects. It's sort of relating brain activity to behavior, high throughput imaging, and then these synthetic data approaches to study movement. Thank you.
Okay, great. Thank you, Tim. Work that you were able to capture in 15 minutes. Anybody want to start off with questions? We'll go to Randy first.
Is the what's the community?
Yeah.
Okay.
It's still on.
Great. Thanks, Tim. Great collection of work. I was just curious about the last part on the modeling for the simulation of the full species, I guess, and whether there's also an opportunity to take, for example, if you're doing stroke research in mice, you can do a stroke model where you actually see behavior deficit, and then see what you would do to transform a motor deficit from a normal mouse to a mouse with some sort of behavioral problems. So I know there's people like, in Wishaw's work, for example, and Lethbridge does a lot of very deep phenotyping of behavior in rodents, where there's an opportunity to kind of work in that domain, use these models to get a better understanding of some of the behavioral variations you may have in animals.
Yeah, absolutely. I mean, that's one of our goals is to better kind of profile the mouse behavior. A challenge working with mice in stroke is that the deficits can be very short lasting, and then you do need to over some of the levels that, that Wishaw has gone to. Another exciting area, I think, will be looking at relationships between the brain activity and the behavior, and see if we can, I guess, better predict, which areas are going to support recovery. So we've tried using different sort of deep learning approaches and GANs and things to be able to try to predict videos of animals from brain activity. And it's been, we've had some success, but I think it's also a problem of models that are overtrained. These are very stereotypic behaviors of mice. And so we can generate a synthetic video of a mouse, of limb moving in response to brain activity, but I'm not sure quite what it tells us yet. But these are directions that we want to certainly go in.
I also like the last part about the application to Parkinson's patients, because I talked to a colleague actually who's going to be doing an interview with Victor tomorrow, Steve Rabinovich. Got a lot of videos of elderlies who are just ready to fall. And the problem with sharing those data, of course, is anonymization. Of trying to make sure that you can analyze the behavior, but not necessarily reveal the identity of the person who's actually falling. And this could be a great way of being able to anonymize those data so they can be actually analyzed in a larger case.
Yeah, that's what we're hoping. We've made some error measurements in our initial publication based on this synthetic capture, and it really does. There isn't too much of a downside. I think we need a larger data set. We need a larger human data set and of affected subjects. And now it's getting into things like, research ethics. How are we going to do this? So most people don't put this kind of thing in consent, that your videos will be shared. And yeah.
Could you tell us a little bit more which brain area you target for stroke?
So we're targeting the motor cortex in our stroke studies, just to see the largest, long-lasting deficit in the mouse, and that is even challenging doing something like that, which would be maybe removal of 25% of the motor cortex.
Thank you for your presentation. How do you build these rodent models? Probably you use existing software, like virtual robotic software or gaming software in which you build the skeleton, and then you start controlling them. How do you do that? How do you control them? Or maybe you can say a few more words.
Okay, so the initial model is done in. So we take the CT scans, and then we get a surface of the mouse from the CT scans, so the volume of the mouse. And then the CT scans also help us to place the actuators. And so those are all done. The surface is done in Blender, and then the bones are manipulated in Blender, and then there's so we can access each joint and then control the rotation around that joint. So we have used in other cases. I know the person who's done it, Louise, has used I think it's called like an NV game engine or something, and
NVIDIA provides some gaming engine.
Yeah. He's used that to actually, so you can use that to control the animal within the modeling environment, and that's certainly a direction to go. But we haven't really exhausted that. The most exciting for us would be to somehow automatically animate the mice, such as what's done with in the human case, where you already have a mesh model of the human body, and then you have a 2D image, and then you generate the 3D kind of best fit of that. And that's how they make those Michael Black and others have that human mannequin image.
But you already have you showed some motor primitives. So
Yeah,
you
yeah.
have controlled your avatars already.
We have, so
how have you
a
done
lot
that?
of that is done, so the animator, so we had a very talented student who brute force, he created a reach. And we also created
Brute force means
brute force
each
animating
somehow the time series or one step after the other.
one step after the other, and then adding things like noise to it, random joint noise. I mean, we also tried things like, you know, crazy stuff like, could we make a library of all possible movements and then move around the joints, you know, have for each one have a range of motion, but we haven't gotten very far.
There are some possibilities to build motor primitives. Yeah, I'm not sure if you were here this morning, but there are some dynamic approaches to build manifolds with flows on it. Yeah, they are often sampled from humans. Yeah, it's an approach that is being performed in human movement science and ecological psychology. So you can actually capture some motor primitives from data and then estimate the corresponding dynamic systems. Yeah? That goes to the degree that you can actually even write characters. Yeah, so capture handwriting primitives or musical motifs playing piano, guitar. Yeah, so these are quite complex movements, and then you can use cortical architectures, cognitive architectures. You can actually use them. These are typically timescale hierarchies to start controlling and putting these motor primitives together. Yeah, then you can start building quite substantial movements, but with a dynamic system approach, which means you have variability in there, you have noise in there, so you can have errors, you can have multi-stability, etc. So this would be possibilities that you can think of, and this you can link to a virtual environment as you're doing already. Yeah? So there may be possibilities to explore. This is also being used in robotic literature. Just ideas.
Well, thank you. I mean, that's what I want to do is you know, learn from this other literature. We just got into it you know, out of COVID, and then once we dig into the human literature, we realize how far advanced it is. But then we also realize that the mouse is a very special case. I mean, I mean, it doesn't walk with 2 legs, 4 legs, and it has you know, kind of a chunky body with a lot of fur. You don't really see the joint, and it's a fur ball with legs. And so it's very challenging.
The Technical University of Munich around Alois Knoll has mouse models that are navigating in virtual environments, and they're
Right.
using also this type of software.
The virtual robot, right?
Yeah.
Yeah, so our approach has been, in our paper, we didn't try to precisely model the mouse, but we used the GAN to kind of cheat and to take videos of real mice, and then so I guess we created a realistic mouse video, but it wasn't really from a real mouse, a model of accurate model. It looked like one. It'd be like a kind of a Hollywood mouse, or you know, just, you know.
Which family of deep learning models were used? Very specific models. They be suitable for brain analysis. We have a limited number of.
we've used deep learning models for pose estimation. So this is deep lab cut, which is similar to deep cut. We have, and I mean, if they're trained on well-trained models, I mean, you can use them on single subjects. In terms of prediction here, you know, I think now, you know, we've the postdoc Dongsheng has worked with GANs, but he's also now gone towards transformer models. But I'm probably not qualified to talk about specifics of doing this sort of brain behavior connections and predictions with that.
Can I make a comment on that?
Sure.
In terms of your automated imaging of your mice where they just enter to get water and get their reward and get imaged at the same time, that would be a way to, as long as you accounted for the autocorrelations between your individual mouse within themselves, that way you get enough data to train mouse model, a model, a deep learning model, and then you could even do it mouse specific deep learning models so that you can account for that variance you described between individuals.
Yeah, that's what we're hoping, is to have round the clock data and then also be able to intervene with things like closed loop, if we can measure from a particular area and try to get the animal to enhance activity in that area and reward it for that.
Good question, maybe with your social interaction recordings that you're able to do. How close do you think you're getting to sort of really modeling social interactions, or is there some level of frustration since they can't around and fight like they like to do?
Yeah, I mean, it's a great question. Like, we first tried this and we were thinking social interaction. We submitted to journals and they're like, you have to think social interaction out. This is not a social interaction. They're on a metal rail. You know? And I don't, you know, I'm not sure what the best experiment to show that it was true. How do we know that they're benefiting socially? I mean, we could look at circuits which are involved in what we think are normal social interaction, interaction between mother pups, or in the ventral hippocampal area, it comes to mind. Some of the reward circuitry, you know, these are things we would look at and then see whether it fits. But does the mouse benefit? Does it enjoy it? I think one way of looking at that would be to have two partners, and the mouse could perhaps make a decision on which animal it wants to interact with, and then we may be able to know something looking at the different brains. So that's what we're excited about, this free mouse interaction. And then, you know, crazy experiment would be somehow an animal can control which partner it interacts with, and we could examine the brain activity. But this is, you know, into the future and crazy stuff. If anyone has, okay, great. Please tell me what to do.
Just coming to mind is to sort of break down, like, what are the signals that are either correlated with certain brain activity or correlated with their preferences. So, like, you could just present them with the smell, just present them with an image, just present them with 3D image. Or, you know, if you could somehow tease out, is it actually the interaction, like the feedback that they're getting from some kind of signal vocalizations? Are they vocalizing when you put them near each other?
So they don't vocalize when they're near each other. Vocalization is a very rare, like ultrasonic vocalization is rare in more around mothers and pups or males and females if they're like courting behavior or something. But we tried and we didn't get anything.
So I have a question. I was wondering if you have considered to incorporate, for example, mouse models of autism and compare how they respond to the same socialization.
Yeah, absolutely. I mean, that's a great suggestion and something we want to do. We're going to start with the Shank3 knockout mouse, which is one of the accepted mouse models of autism. Would be a possibility, and then but I think we first need to get that paradigm to establish that really what does social interaction look like, and how do we measure that the mice are really benefiting from it and they like doing it, and so that's definitely down the road. And then once, if we could identify some kind of aberrant or different pattern of brain activity in lines of mice that were associated with human autistic characteristics, then we could try a corrective strategy to change brain activity. And that's one of the things we want to do with some of the deeper subcortical circuits is, can we train animals to increase activity in a particular cortical area that may have a beneficial effect on emotionally salient circuits? I don't know if it will work, but that's a goal.
Be really interesting.
Okay. Any more questions? Okay, if there's no more questions, that puts us on time, actually. We're at a great session.